Okay, hello everyone. Welcome to this new seminar in Ecology and Evolution of Montpellier. Just a few technical words for those of you who are online. As usual, you are in a webinar mode, which means that your microphones and cameras are automatically switched off. If you want to ask questions to today's speaker after the talk, you can use the Q&R tab to ask your question. And then we will switch on your microphone and camera such that you can ask your question directly to the speaker. And if you have any technical issue, please use the chat to report, and I will try to help you to solve the problem. Okay, and after this, I give the floor to Florent Mouliot, who will introduce today's speaker.
Everyone, thanks for coming. So we are hosting today Colin Prentice. So he's visiting us at CEF as part of the Fire Adapt project led by AMAP. So Colin Prentice is a professor at Imperial College in London. He's also the director of the Leverhulme Centre for Wildfires since 2019. His career has been started in Lund and Vienna, where they have been developing the early stages of dynamic global vegetation models, simulating the exchange between biosphere and atmosphere, including vegetation dynamic. Then he moved to Australia, and he got a chair, an AXA chair on biosphere atmosphere interaction. He's now running an ERC project called REAL, which is the Reinventing Ecosystem and Land Surface Model. So that's what he's going to talk about today. With the concept of switching from process-based models, more based on ecophysiology, to more theoretical advances on trait selection. So, Colin, the floor is yours. Thank you.
Thank you, Florent. I guess you know this, but we can start here. The land surface is not just a passive recipient of changes in the atmosphere, but is actually a major actor itself in determining climate, the nature of climate, and of course carbon cycling. So on the climate side, transpiration, in other words, the flow of water through plants from the soil into the atmosphere, constitutes about 70% of the total water that is evaporated from the land surface, the evapotranspiration. And that transpiration is very tightly regulated by plants. So the key quantity is the conductance of the canopy, which is regulated by the behavior of the stomata, which are the tiny pores in leaf surface, which let carbon dioxide in and water out. Gross primary production is the name we give to photosynthesis at a large scale, and it amounts to about 150 petagrams of carbon per year. It's the largest flux in the global carbon cycle, and just to put things in perspective, the total emissions from fossil fuel burning is only about 10. So you've got this flux going on all the time that is massive, even in comparison with the fossil fuel emissions. All climate models, therefore, now at least all modern climate models, include a land surface model that somehow represents the behavior of the land surface. And all modern ones represent this coupling of CO2 uptake and water loss through the stomata. And many of the current models also include explicit representations of carbon cycling and vegetation dynamics. Now, the term, this term third generation, this has been around for a while, and in fact, we're still in the third generation, basically. It's since 1997. It was a breakthrough article by the late Piers Sellers, who perhaps gained greater fame as an astronaut, but he was an Earth system scientist. And this is one paper where it's like, I remember where I was when I read it. I remember exactly where I was and what I was doing, because, you know, here was the insight that would lead to a proper scientific basis for land surface model. So, and the basic insight is extremely simple. It says that photosynthesis has to be, at any one moment, it has to be equal to the product of the gradient of CO2 across the stomata. So Ca minus Ca, so Ci, so carbon dioxide, partial pressure outside the leaf, and the difference between that and the carbon dioxide partial pressure inside the leaf, which of course is being drawn down by photosynthesis. So it has to be proportional to that, and the proportionality is the quantity we call semiconductance. So that has to be always true. And at the same time, the transpiration, which we use with the simple symbol E here, the transpiration has to be proportional to the water vapor gradient, which is basically saturated inside the leaf, unsaturated outside the leaf. The difference is the vapor pressure deficit, or D, and again, they're linked by semiconductance. The factor 1.6, just because water and CO2 molecules are different sizes, so they diffuse at different rates. You can treat that semiconductance, you can measure it at the leaf level, or you can just treat it at the whole canopy level. You can treat the canopy as a so-called big leaf, and that was another innovation that Piers Sellers introduced explicitly. It's remarkable how well this works. It's not perfect, but it does about 95% of the job and makes our life simple. So what about these land surface models? Well, there are some problems. In fact, there are some big problems. One is that current land surface models, when they're asked to make predictions under future scenarios, even if they all get the answer about right for the present, they go off in all directions. So they predict different futures for transpiration and carbon uptake. If you look closely at what's in such a model, you'll find that often the representations of specific processes are okay for the fast timescale, the sort of timescale where we have measurements and models based on measurements in the lab, but on ecological timescales, or even from sort of weeks onwards, quite often their process representations are not good. They're not good at treating the processes by which plants acclimate to changing conditions. And I'll say a lot more about that later. The models are also surprisingly complex, considering the simplicity of the basic concept. And what's more, they're getting more and more complex. And because they're so complex, they're not transparent. So if your model is not doing well, it's almost impossible to find out why. You can't fix it because this thing has been developed by generations of postdocs and so on. It's a model with a name. This is the model they use at a particular lab. It has some code that's been written 30 years ago in an obsolete version of Fortran. Nobody can make any sense of it. I caricature it a little, but not much. And perhaps most importantly, the connection between the effort of making a land surface model, which is now mostly done in climate modeling centres, there's very limited connection to ecosystem science. So a lot of papers, some of you will have written these papers that point out that it's important to study plant function because it's going to improve ecosystem models. But in fact, it doesn't, because the ecosystem modelers are one lot of people and you're another lot. So what I mean by limited connection, people who work on land surface models, they certainly read papers on this subject. They read, new phytologists and global change biology and so on. They read the papers, they don't read them critically. So what they do is they accept the conclusions that they find in the literature. Turns out that's a bad mistake. Here's just one illustration. I could give loads of illustration with how terrible the models are, but I got fed up with doing that. So just going to show one picture here from a paper in preparation led by Benny Stocker. Just look at the left-hand diagram here. And on the right, you see the actual carbon sink. So this is the carbon uptake, which we can measure by reasonably well by various methods at a global scale. So that's the actual uptake, net uptake of carbon during this period. It's something that's been ongoing ever since CO2 started rising. But that's the net uptake of carbon. It is known rather accurately. And in the middle, you see models that are so-called carbon-only models. So what that means is they do model carbon and water cycling and energy exchange, but they don't model the nitrogen cycle. So they're called carbon-only models. Now, about 20 years ago, the modeling community became obsessed with the idea that they needed to model the nitrogen cycle, because they had the idea that we were greatly overestimating the capacity of the biosphere to take up carbon because of ignoring nutrient limitations. Anyway, those are the carbon models, and these are actually from recent compilations that include models of both types. So you see carbon-only models. Well, actually, they're quite good at simulating the actual observed carbon uptake. There's a bit of a scatter around it, but it's not a very big scatter. So you might say, well, why was everybody so worried? Well, I do wonder sometimes. Anyway, here is what we have now with the new generation. You know, after 20 years of development, we have coupled carbon nitrogen cycle models. And look, they slightly underestimate the thing instead of slightly overestimating it. But the actual carbon uptake is well within the range because the range is huge. In other words, the uncertainty has massively increased. And of course, this is because although everybody knew that they should make a model with a nitrogen cycle, but nobody knew actually how to do it. So there are all kinds of methods and all kinds of fudges and things brushed under the carpet in these models. Okay, so I'm complaining about models. Now I'm going to complain about ecosystem science. The main problem here, in my view, is the lack of a coherent theory in ecosystem science. There's a lot of ideas that are knocking about, but they're rather vague. And as a result, I argue that we see the persistence of a number of incorrect beliefs. And in order to be maximally provocative, I've made a list here. This is by no means a complete list of things that I maintain are incorrect, but are believed by many people in the ecosystem science community. And I'm not going to talk about all of them, but I will. At the very end of the talk, I'll talk about the last one there, about nitrogen limitation. Now, I think the problem is compounded by the standard research methods. And here we have to sort of go back into the origins of ecosystem models, because when it was realized that global change was, you know, something we should seriously be concerned about, then it became clear that we needed models that were, you know, global in scope, but were able to represent the key processes in the carbon and water cycles. And ecology was really, as a science, was really not prepared for this. If we go back to the 1980s, there was absolutely no theory that could really be used. So it's been an uphill struggle, but of course it's been a big there's been a big expansion in this field. But there were very, very few data, for example, and that made it kind of tough to evaluate models. So what we have now is a situation where in land surface models, there is a standard method. This really is the normal thing that is done. You make a complete model of the global land biosphere, and you run that model, and you put in, either you change a process, or you put in, very commonly, put in a new process that you didn't have before. And you then compare with some sort of benchmark data. You say, oh, look, we got an improvement. But what people don't do is go back and test all of the individual processes in the model. In other words, they benchmark the whole model. They don't go back and analyze the way in which they represent fundamental processes. And I believe that's a big problem. On the other hand, in ecosystem science, many, many papers gather data sets, and of course there are now tremendous data resources available, but they analyze data sets looking for causality, except the only thing that they would ever find is correlation, and the same applies in space. If you use machine learning, you will never find causality this way. What you will find is the prejudices that you put into your analysis. So if the results are consistent with what you thought before, you will say, well, this shows it's consistent with what I thought before. So my argument is neither of those research methods actually works, and we should adopt quite a different one, which is as follows. We should formulate explicit hypotheses, quantitatively, of course, and then we should use the available data to test the hypotheses one by one. Now, it has often occurred to me that we have a name for this. It's called science. This is the way that science is supposed to be done, and I think this is the way we should do it. More specifically, there is a concept which I and many collaborators have used extensively over recent years, which is called eco-evolutionary optimality. And at step one here, this really helps. You know, what hypotheses can we have about how plants work? They're very complex items, there's a huge diversity of plants and so on, both within and between regions. What do we do about it? Well, there is a principle which can be argued to be simply the central law of biology, which is that natural selection rapidly eliminates any trait combination that results in inefficient use of resources, or more precisely, that could be, you know, where the use of resources could be done better by another species that is competitive. And it's not a magic bullet. I mean, there isn't a formula that says, okay, eco-evolutionary optimality says, you know, this must be so. There are many constraints. There's questions about what plants can and can't optimize, or what species can and can't optimize. And exactly what the criterion that is optimized, we don't know. But it's very useful because this enables us to cut through a lot of complexity and come up with hypotheses which can be tested. And if we got the wrong criterion, then the data will tell us. And of course, at step two, we now have fantastic data resources. Again, if we go back to the 1980s, we just didn't have anything that we have now. We didn't have flux towers and remote sensing data, and we didn't have trait database. You know, we didn't have any of the stuff that we can now rely on to do this science. So I'm going to go through a number of examples in varying degrees of detail. I'm going to give most detail in the first case because it's relatively simple, and the theory that we're using here has now been around for 10 years. And obviously there were earlier theoretical developments dating right back to the 70s. This is about the ratio of carbon dioxide inside the leaf to outside the leaf. That's a very fundamental quantity, because in addition to those basic diffusion equations I showed earlier, empirically, it appears that this ratio is maintained at remarkably constant values over a range of environmental variation. Particularly with CO2, it changes just a little, but only slightly, with fairly large changes in CO2. With light, it doesn't change at all. And we've known that from experiments, actually dating back to the 1970s. We have also known that as you increase the vapor pressure deficit, and this happens fast, as you increase the vapor pressure deficit, that ratio goes down, which is intuitive. In other words, if water becomes, you know, greater demand for water, the stomata close up a bit, and so they let less water out. And from the diffusion equations, we can write an expression for the stomata conductance in terms of this ratio, which I call chi. And these are the equations that are used in land surface models. Most of them use one of these three. They're always expressed as equations for stomata conductance, but they're easier to understand if you define them as expressions for the ratio chi. So here we have the Wall-Berry model, which has one parameter. Luning model has two parameters. Medley model has one parameter. But the way that these are implemented is because those parameters empirically seem to vary, therefore they are assigned a constant value for different plant functional types. So the idea is we divide the plants of the world into a few, about 5 or 10 plant functional types, and we declare that the parameters here are going to be constant within each plant functional type. Now that is a big simplification, but it's probably better than assuming that their values are the same everywhere, which would lead us into big problems. But it's not a very good solution, not the best solution. This is a much better solution. So we make an explicit hypothesis, which says that the plants minimize the sum of two costs. And one of those is the cost of maintaining the pathway for transpiration. And the other is the cost of maintaining the biochemical capacity of photosynthesis, which is expressed by VC max. And you've got A on the bottom there that says that these are unit costs, so these are costs per unit of CO2 assimilation, CO2 uptake. And we've got two parameters in there, which we have imaginatively called A and B. So A is basically the cost of maintaining the water pathway, which is necessary for photosynthesis. And then B is the cost of maintaining the biochemistry of photosynthesis. And to cut a long story short, very simple. standard, simple, very, very simple calculus, and together with elements that we take from the Farquhar model, which is the standard model of photosynthesis, leads us to a formula that can be approximated like this. Now, this appears to be identical to the Medlin model, which was derived using a different criterion. If we just go back one here, you see the Medlin model. In fact, the only difference there is that Belinda Medlin hates Greek letters, whereas I think they're quite fun. So I put a Greek letter in there, which I call squiggle, properly called psi, which is the parameter governing somatic behavior. But now we have an explicit expression for what this parameter is. So it's not just a parameter that we have to assign for a plant functional type. It can be expressed in terms of the known kinetic properties of Rubisco and one parameter, which is the ratio of these B and A costs. And that we determine empirically from a lot of data on CI/CA ratios. But we already know from first principles, we expect this B constant to be constant, B parameter to be constant, but A we expect to decline quite steeply with temperature because water flows more freely at high temperatures, and it's really quite a big change in viscosity. And this is a somewhat neglected aspect of plant biology. It's very important if you're taking a global perspective. So there are certain consequences we can predict. From that theory, we predict, of course, that this ratio goes down with vapor pressure deficit, as is known to be true. But we also predict it increases with temperature, and that's because the Michaelis constant of Rubisco goes up and the viscosity goes down, both of which are pushing up this ratio. And it goes down with elevation. And that is due to the partial pressure of oxygen. I won't get into the details here, but this all follows from the Farquhar model, and also the vapor pressure deficit, which, other things being equal, does go down with progression. So turns out all these things are correct. Now, the vapor pressure deficit response is a fast response and has been known for a long time. The temperature response is not known at all, even though it's really large. However, it can be shown if you actually do a transfer experiment. So you're growing a plant in a pot in a controlled environment, and you move to a warmer environment, then sure enough, that ratio increases. There's a paper here that showed it. The paper actually misinterpreted what they saw, but nevertheless, this is a critical paper that shows exactly this phenomenon. It's been shown again in a few papers. The elevation response is interesting because it has been known for quite a long time. And it's interesting to go back to the papers, so they realized it was something to do with the partial pressure of oxygen, but they didn't have a complete theory for it. So now, anyway, it just falls out nicely from our theory. So now the point is that this is quantitative, right? It's not just directions that are correct, the numbers are correct too. So we can actually, we can make a prediction of the slope of, it's actually logic transform of chi. We use a stable light carbon isotope data on leaves. We have data set with thousands of leaves in it across the world. And so we can actually do empirical analysis. We can do multiple regression to get the partial effects fitted from the data. And we can also make theoretical predictions. And as you can see, they are actually in agreement with one another within the error limits of the data. But nothing like this has ever been shown before with plant traits, right? It's always about directions of change and so on. So here we can actually show that we get the numbers correct. Here is a more recent application from Adrian Lavigne's work. This is actually tree ring. So each of the little sticks is a tree ring series. And you see here the partial effects of logic transform chi against temperature at the left there, increasing the temperature, decreasing the BPD, slight decrease in CO2, and decrease in elevation. And the red is the prediction, the black is fitted from the data. And you see that we predict all four of those relationships correctly. So that was an example which shows what can be done. And I think we might be lucky with our optimality hypothesis, because it isn't the only one that can be formulated. In fact, there is a much older one in the literature which was used to infer the measuring model. But the point is that our hypothesis turns out to, which is was originally described by Ian Wright, but in not such a quantitative way, about 10 years before. But that hypothesis actually correctly predicts all of the observed aspects of how this ratio depends on physical environment. Okay, so I'm going to go on a little more briefly to discuss some other cases. Because having got that ratio, because we need a few other things in order to make a full model, even just of photosynthesis. So we want to know now what controls photosynthetic capacity, or VCMAX, which is basically the activity of the enzyme rubisco that actually does the carbon fixation. And here we have a very simple hypothesis that also has quite long antecedents. It's been suggested many times in the literature, which is that this value, which is regulated by the plant, so this is the activity of rubisco in the leaf, the value should be just large enough to use all of the available light. No more, no less. Because if it was less, then another species could come along that would grow faster and outcompete the inefficient species. On the other hand, if VCMAX was any higher, well, there is a cost in respiration, maintaining enzymes in an active state. And so if it was more, then there's a wasteful use of energy. So there should be a co-limitation by VCMAX and light. So it's an old idea, but it had many implications that have all been missed. So here are the explicit consequences. We should predict that VCMAX will increase actually in proportion to light. So if you double the light, you double the VCMAX. And it turns out to be quite true. It should increase with temperature, but the increase is not the increase that's determined by enzyme kinetics. You can measure in the lab, you can measure the enzyme kinetics in the lab, and you get a steep increase with temperature like that. But the plant is not so stupid as to maintain that same the same amount of rubisco if it gets warmer. So the base rate goes down, and the result is you do still get an increase, but it's a much less steep increase than you get from enzyme kinetics. So all the models basically assume it's just the same as enzyme kinetics, but it's not. And so the result is if you standardize this to 25 degrees, which is commonly done, then it goes down in temperature. And this is just showing, just focus on the top row, a large global data set. So here standardized 25 degrees, we see the response to light on the left and temperature in the middle. So this is, the growth environment, light in the growth environment. It's not just what we shine at it, it's the actual light that the plant experiences in its natural habitat. And the growth temperature, and then also an effect of vapor deficit, because as the stomata close, you need a bit more enzyme in order to use the available light. So, there you see the responses. That's just showing the direction, but we can also, again, compare theoretical values with observed values. So the log slope should be 1 for light, which it is. For the temperature, for growth temperature, it should be minus 0.05, which is within the error limit, and a small response to vapor pressure deficit. And there are now ways in which the VCMAX can be seen from space indirectly, but by the chlorophyll content. And so this is a remotely sensed map of VCMAX at the top. And then this is a prediction based on our Ecologie-Evolution hypothesis. Now, of course, we can go further and we can look at the effects of CO2. And so here is a comparison with a meta-analysis, quite an old meta-analysis, but the numbers basically haven't changed in the meantime. So this is showing how light efficiency changes in experiments where CO2 has been increased, how water use efficiency changes, and how thermal conductance changes. So you see light use efficiency goes up, and water use efficiency goes up a lot, and thermal conductance declines. And we're consistent. Our predictions are consistent with the meta-analysis. I missed that one step here, which is called Jmax, but I'm not going to go into that. That's a refinement. But the point is, we've got VC-max, J-max, and the CICA ratio. And we've got satellites up there measuring the green vegetation cover. We can put the pieces together and make a prediction of gross primary production. Photosynthesis every half hour across the entire globe. And we can do this with flux sites, ecovariance flux sites, where the CO2 uptake is actually measured, so GPP is actually measured. And so this is like, you know, every half hour during 1 week is the same week. You look at the daily cycles at 5 different sites in completely different environments. And the grey traces is the error range of the GPP measurements, and the red traces are the prediction from a model that is based on these various hypotheses. And the key point here is that there are no plant type specific parameters. There is just one set of exactly the same set of parameters that is used to generate these graphs in each ecosystem. And the point is that it does equally well. So it's like if you have a model that has a whole long list of parameters for every plant functional type, and you've got 10 plant functional types, that's a lot of parameters. But the point is you don't need all those different plant functional types because one model will do the trick. Okay. Now, what if we're not relying on satellites? So suppose we want to predict things that have happened in the past, or we want to predict what will happen in the future. So we're not relying on satellites then. we need to predict how green the vegetation cover is across the world as a function of time. A couple of key concepts here. FAPAR, the fractional absorbed photosynthetic active radiation, that's basically greenness, which can be measured in various ways, but all of them ultimately rely on the absorption of red light by chlorophyll. And then the LAI, which is a kind of a biometric concept, projected leaf area per unit ground area. And the idea is that these two are reasonably well linked through Beer's law here, which just has this one coefficient in it, K, the licensing coefficient. So we have a theory now for what the greenness should be. And it should be the lesser of 2 quantities, one of which is limited by energy and the other by water. So here is the energy limited version. We write a very simple expression here for the net carbon profit. So you have A naught, that is the potential gross primary production if all of the light were absorbed by the leaves. That is then multiplied by this expression from Beer's law. And then we take off a certain amount for the costs of constructing, maintaining leaves, and the roots that are needed to supply. And it could very simply be shown that there is a maximum, provided there's enough water to do this, there is a maximum which can be expressed as a simple formula for either LAI or FAPAR. But there might not be enough water, and in fact, water is the limiting factor for primary production, the limiting factor of most of the world. We can write a different equation which says the transpiration has to be equal to the water use that is demanded by photosynthesis modified by the green vegetation cover. Because if you have a dense vegetation cover that is, transpiring at a potential rate, it's going to run out of water very quickly in a dry environment. And we make a very simple assumption. We assume that there is a parameter called F naught, which is the fraction of total annual precipitation that can be used by plants. We're hypothesizing that that is a fairly stable number, and then we can write an expression for what is the FAPAR that could be sustained given the annual precipitation. Hello? Well, that's a pity.
Try and press it. Yes.
Oh, oh, you made some animation. I didn't put an animation. Well, anyway, never mind. Surely put an animation in here. Right, okay. Yeah, that's fine. Good. I thought we were missing something for a minute. Okay, so there we have modeled and observed seasonal maximum FAPAR. Obviously, not all leaves are displayed all the time in every ecosystem, but here we just look at seasonal maximum because, of course, enough carbon must be allocated to make those leaves and enough water must be available for them to photosynthesize while they're there. So we have a modeled and observed field, so the observed derived from MODIS data and model derived from our model, and the little inset at the top is showing the energy limited regions versus the water limited regions of the world according to our calculations. And here, this is just a comparison. We put in here the so-called models from the so-called trendy model is comparison exercise, which is carried out every year, just showing, all the models can do this, the first order. The statistics are actually slightly better for our model than they are for any of these much more complex models. But the most important thing is that our model is really simple. There's no, we haven't got 10 PFTs or anything. We've got much shorter list of parameters. And almost all of those are estimated independently. So there was a couple of parameters in that model. There was the so-called Z cost of leaves and the F naught, which is the fraction of precipitation that can be used. We've simply assumed that those are globally constant and estimated them using this observed map. So there are two parameters in there that are estimated from data, but only two. So then we can look at trends again. It's from work of Ziqi Zhu in Tsinghua. And what we're looking at here is the MODIS FAPAR trend and then the GPP trend that we infer from it. And so PL model here means this means that we have simulated both the green vegetation cover and the photosynthesis that results. So this is the green vegetation cover on the left at the bottom, and then at the right at the bottom is the simulated gross primary production. You see there are increasing trends in both, and those increasing trends, you get the magnitude is slightly different, between our model and the data, but the trend is the same. There is an increasing trend, and this is our most recent version of the principal drivers, because once you can simulate it with the model, you can find out what the drivers are, at least according to the model. So blue is pale blue over much of the world. That says the trend mostly driven by rising CO2, but the yellow part in the high latitude says it's mostly driven by the lengthening of the growing season. And then there's a few other controls in there. Some areas precipitation has been increasing during this interval, like the Sahel, for example. So the greening there is influence is driven by the increasing rainfall. So that was the seasonal maximum FAPAR. What about the annual time course? We'd like to be able to simulate that too. And I'm going to gloss over a lot of details here because this one is quite complicated. But the main point in the workflow at the top is the part where I put a red ring, and this is a theory which we didn't invent. It's a new application of a theory that was developed a few years back, published in James. The basic idea is that the phenology of vegetation will also follow an optimality rule. The rule is that at a time that leaves will be displayed at the time or close to the time when they have the most potential to photosynthesize, which to me makes a lot of sense. And the key thing here is the definition of a concept called the steady state LAI and the steady state GBP. And the idea is that these should be in equilibrium with one another. So what you do is you consider what would happen if the weather conditions this week or month were continued forever. And there should be an equilibrium. There should be a GPP that will be sufficient to produce a certain amount of leaf area, and then the leaf area that will be sufficient to allow that amount of photosynthesis. And this does actually have an explicit solution, which is based on the Lambert W function, which is very convenient for this purpose, and actually gives us a modeled time course of LAI. And here I'm only showing one sort of graph here, which just shows that with the help, there are many steps here, of course, but with the help of this model, we can simulate. This is not the annual data anymore. This is monthly data. We can simulate a monthly time course of leaf area index. Here I haven't shown you the result of comparison with all the trendy models, but in fact, this is a case where our model is not just similar, it's actually very much better than all except one of the trendy models. So it's another story. Okay, so far it's all been about photosynthesis, but plants also have to respire. So here we're looking at the acclimation of leaf respiration, and there are three columns here: dot respiration at the left, photosynthetic capacity in the middle, and their ratio on the right. At the top is quantities measured at growth temperature in a global data set, and at the bottom is standardized at 25 degrees. To cut a long story short, the response to respiration is very similar to that of VC-max itself, such that when they're standardized 25 degrees, the ratio is completely flat, which is good because that is what people have actually quite commonly assumed. Turns out this is correct. But here you see, for example, you see that the respiration of leaves, look at the top there, because it does increase with temperature, but it increases more slowly than the dotted line, which is the enzyme kinetic rate. All models basically assume the enzyme kinetic rate, but they're wrong. They're overestimating the sensitivity of respiration. And this is a similar analysis by Zhang Han at Tsinghua on a whole lot of data which he had to assemble from literature, a whole lot of data on actual measurements of sapwood respiration, both in situ and ex situ. And the key point that is shown here is that with the growth temperature, whether the measurements are made in the lab or in the field, there's an offset between them. But the point is that with growth temperature, increasing growth temperature, the standardized respiration rate and even the actual growth temperature respiration rate shown on the right actually decline. So in that case, again, you see the grey line is what models assume, which is they assume that stem respiration increases in temperature, and this is what the observations show, which is that it decreases in temperature and at a rate that we predict. I'm going to have to go very fast through the leaf economic spectrum. I'm sure a lot of you know very well about this. A ubiquitous negative relationship between two critical quantities of the leaf, which is the leaf mass per area, so how thick or dense it is, and the leaf lifespan, in other words, how long it stays on the tree or the shrub. Now, we know there is this negative relationship. We also know the relationship shifts with climate. Now, there's quite an old model here. It explains it. It's the first order. That's this one. And we write an expression for net carbon gain, which is up top there. So this is from Kikuzawa's original work, simplified a little bit. The idea is that the integrated, if you look at the integrated photosynthesis over the lifespan of the leaf, and consider then the construction cost of the leaf, and amortize the results over the leaf lifespan, you get the net carbon gain. And that should be maximized. Again, maximization leads to a very simple expression for the net carbon gain. And it turns out that you can have a lot of different values of leaf mass per area and leaf lifespan that all satisfy this criterion. And that's why we have a leaf economic spectrum. But we can go further. These are predictions that are directly made by the Kikuzawa model, and it includes term which is called B here, which is the inverse of the aging rate. So the idea is that the longer the leaf stays on the tree, the more it loses photosynthetic capacity. What controls that? Isn't that also controlled by LMA and by photosynthetic rate? And the answer is it is, according to this model, supported by data here, showing that after an initial stage, there is a gradual decline, a linear decline in photosynthetic capacity, and that can be expressed, aging rate can be expressed in terms of LMA and BC-max25. So we have, well, I showed you some data on this before. We can put all these things together, and in the end, we get a single expression, which tells us about the relationship between the optimal lifespan and the LMA. And we can apply this. Here it's applied to a whole lot of data, for which we actually have leaf longevity. So here's a very, on the left, very straightforward relationship between LMA and longevity. But the more bright the environment, the shorter lived for a given LMA. The fraction of the growing season comes in here. These are all evergreens, the first three panels, and then the one on the right is deciduous plants. And deciduous plants play by a different rule, because their leaf lifespan is basically dictated by the length of the growing season. We had to resort to a new trick here. So how do you predict the optimal value of LMA when all you know is a relationship between LMA and leaf longevity? Usually you don't have the data on leaf longevity. So we decided to work with what nature gives us. So we start with all of the data we have from all over the world about LMA and leaf longevity. We make statistical distributions of them as represented by that bottom right graph. So that gives you these concentric circles. And we then put a line through. The line is our optimum line. And in different environments, the optimum line occurs in a different place. And so if you start with the prior distribution, which is given by those circles, you put a line through it, then you get a normal distribution. And that has a peak and standard deviation. And that's what we do here when we don't have leaf longevity. So this is from a more extensive data set. We're just trying to predict the optimal LMA. And this is done for evergreen plants at the top and deciduous plants at the bottom. And our predictions are shown for light and then temperature. There's also an effect of aridity, which is not really formally part of our theory, but we do show strong effects of aridity, and then fractional growing season length at the end. And then we can predict some things that are observed. Here you see solid lines. What this is looking at is the trend in the equator to pole gradient. So as you go north from the equator, you find that evergreen leaves get thicker and live longer. And as you do the same with deciduous plants, you find the opposite. They get thinner, and they live for a shorter time. So what you see is data there with absolute latitude, and you see the predictions, which are the dashed lines, which you see are almost exactly coinciding with the solid lines. And finally, so this is the point about nitrogen limitation. There's been a lot of fuss in the literature about declining plant nutrition. So people are measuring, going to a various specimens showing, oh, look, modern leaves have less nitrogen in them than leaves of, 50 or 100 years ago. And it turns out that yes, so they should have. That's exactly what we predict. We predict that there should be a declining leaf nitrogen due to CO2, which is shown in red, due to warming, which is shown in blue, and then the combination shown in black. The decline is at a rate of about 0.27% per year. Literature reports are between about 2 and 2.2 and 0.25 per year. There's nothing to do with nutrient supply. It's just because the plants are smart and smarter than they're usually given credit for. So there's no problem here. The plants are just doing the thing that they always have done. Okay, just some acknowledgements at the end. So yes, the grant is my ERC advanced grant, but also a lot of this work more recently has been funded by Schmidt, or it's now called Schmidt Sciences, the Elementary project, which is an international consortium. The lead PI is Sandy, who's sitting here. And this also, a lot of the results I've shown today are from the work of the group, not only my group in Sandy's, but also the group of Han Wang in Tsinghua University, who is one of the 11 also co-PIs on this large international grant. And there's some literature. Okay, right, I'll stop.
Very much. We will move to questions.
Thank you for your talk. I remember there were some studies suggesting that, okay, you mentioned that stomata conductance is heavily controlled by the behavior of the stomata, opening or closing. I remember that there are some studies suggesting that stomata conductance is also influenced by the ecological adaptations of the leaf, including stomatal frequencies or stomatal size. And I wondered what would be the relevance of these studies in the context of your modeling?
So the thing that matters from the point of the plant function, of course, is the stomatal conductance itself. And changes in stomatal conductance can be brought about in different ways. And there have been a number of experiments where people have looked at changes in stomatal density with CO2, for example. And some plants show a change in stomatal density and others don't. I mean, with the same plant. But then, we can look beyond that to differences in species. And then if we go back into the geological record, we can go back to times when CO2 was very much higher. I mean, far higher, like 2000 ppm or something in the Eocene. And there, of course, they're not the same species as we have now, but there are lots of leaf records, and the leaf fossils are good enough to count stomata. The stomatal densities are incredibly low. They have very, very few stomata. So that was obviously, that was the way of, plants at very high CO2 just don't need to have high stomatal density because they don't need a high stomatal. That's the way I behave.
Okay, thanks for the great talk. Can you hear me well? Can you hear me?
Yes.
Yeah.
Yeah.
Okay, sorry. So thanks for the great talk. I have a question about VC max values. So looking at your plots, actually there are lots of variation for given growing conditions within a site, and we know that. And those variability within a site might be important to account for local community dynamics and projection of floristic shift. So I would like to know what would be your recommendation to infer VC max variation within a site.
Yes. My understanding is that the major cause of VC max variation at a site, which of course can be quite large, is simply the light climate. That's well known. I think that VC max in the understory is much smaller. So if you're going to make, we were just looking at very simple way, look at pulling all the data from a site. But if you're going to make a dynamic model, you certainly would need to take account of the vertical gradient of VC max. You could predict that, at least you could try to predict that just from our theory. I think you would probably overestimate the steepness of the gradient. So there is an issue there that we're looking at, which is that the extinction coefficient of VC max is much smaller than the extinction coefficient of the light itself. We think the reason for that is that plants in the understory are able to get, they get sun flex. So they can, some of their photosynthesis is done at light levels are much higher than their average light level. And I think that probably is the explanation for why the VC max in the understory is greater than our theory would predict. But that's partly speculative at the moment. We need more data on it.
Thanks for the talk. This is not my field, so I might say some mistake. This is also about VC max. You showed us how temperature should affect VC max according to the optimality criteria of your model. Do you know what would be the effect of rising CO2 concentrations? Because I guess a VC max is affected by the affinity of Rubisco for CO2 too?
Yes, yeah. So there we know. Yes, we know exactly what happens there, which is that VC max declines as CO2 increases, and that's what was behind the very last graph that I showed. So this decline in leaf nitrogen is a result of declining VC max, and it's just because it's because because the Rubisco is not saturating. CO2 is not a saturating level now. So if CO2 increases, then less VC max is needed in order to use the available light.
I think it was a rather fascinating the response of I would say decreased quantity of chloroplast, I would say, when you have CO2 increasing and things like that recently with temperatures. This seems to be a plastic response. Can it be anything else than the result of competition among chloroplasts within cells as a short-term response, which leads to all the series of responses? Because this cannot really have been selected in itself, the curve.
I don't know about the mechanism. And you could ask questions like that about many of the things that we show. You can ask how does the plant do that? Now I don't know. I'm not a plant physiologist or anatomist, but it is an interesting point that what we're actually not trying to do is to simulate the mechanism. We're trying to simulate the outcome. Right. Now you can ask questions like about everything that we show. It is an important question. Are we looking at a plastic response, or are we looking at some, genetic differences between genotypes or between species? And the answer turns out to differ, because we can do the trait gradient analysis where we're looking at variation in traits both within and between species along gradients, and we find that the answer is different for different traits. So, for example, the CI/CA ratio is almost perfectly plastic, whereas if we take another trait like leaf size, for example, is almost perfectly non-plastic. Has a slight plastic response, but is more or less constant per species.
Yeah, the read question. So all your rules you fixed here, they are across species. So you have the same rules according to plasticity within species, or do you think it's completely different?
Can you say that one again?
The rules you proposed here are mostly for across species.
Oh, okay. Yeah, no, no, that relates to my last answer. The point is that the plasticity of different traits is very different. So the CI/CA ratio, we can just throw in all data together, and we don't care whether it's between or within species, because the response is the same. But if we look at LMA, the patterns that we show are dominated. As far as we can see, they are dominated by species replacement. And LMA is less plastic; it's intermediate. But on the other hand, there are experiments that show that LMA adapts in or acclimates in a similar way, qualitatively, to what we predict, but with less magnitude. Now, it wouldn't automatically be true. I imagine, there might be cases where there's between species response was qualitatively different, but we haven't seen anything like that. It seems that there are limits to plasticity of some traits, but nevertheless, insofar as they can adjust within a species, then they do so in a similar direction to what we see.
Okay, all right, very good. I will give you back to that.